Okay, we're going to start the first afternoon session. And I'm very happy to have Luisa Cutillo. She is an associate professor in the Department of Statistics at the School of Mathematics, University of Leeds. Yes. Thanks for being here, Luisa.
I guess I'll start. Can you hear me? Just to make sure. Yeah, perfect. So, hi everyone. Thank you for joining this afternoon. And I'm happy I'm after lunch, so you should be all relaxed now. And I usually have a nap after lunch, so I hope you, you won't fall asleep during my talk. And today I'm going to talk about Gaussian graphical models in multiple omics data. And I will show you some theory and some applications. And for the application part, we will focus on a methodology that we have been developing together with Bailey, Andrew, that is here, is the third year PhD student, starting now his third year. I wrote Andrew Bailey, but Andrew is his surname, sorry. It's Bailey is his name and Andrew is his surname. I always do that. Just, it's not my fault, they look so similar. I have to switch, I'm so sorry. Okay, and the other supervisor is David Westhead, that is a professor of bioinformatics in the University of Leeds, where we all come from. But Bailey is actually based in computer science. I am in maths. So the talk, oh no, it's not the wrong, it's the wrong part, it's here. Okay. The talk is divided in 3 sections. So just to set the expectations, we will go through the background on Gaussian graphical models. Then we will talk about our approach. And at the end of the talk, we will give a live demonstration on how to use our methodology. So we have prepared a code space page on GitHub. So I hope all of you got the GitHub account. If not, it's a very bad thing. You should all have one. Okay? Great. So, background, part 1. What is a Gaussian graphical model? So it's a fancy name for a multivariate Gaussian distribution in RP. So it's completely specified once we have the mean and the matrix of covariances. Okay. So, this image is just easily taken from Wikipedia. There's 2 dimensions there. And the number of features we will refer to often into this, in this talk would be this number of variables that are involved. And another main player of the talk would be this matrix, that is the inverse of the covariance matrix, that is called precision matrix. Okay, so why do we say then graphical model, Gaussian graphical model? So a graphical model can be associated to any distribution, and here there's the underlying, as we were saying, is a multivariate Gaussian distribution, but the word graph is associated to the structure of a graph that needs to be defined. So in our case, the vertices of the graph would be the random variables, and the connections between these random variables would be the edges. Now, what kind of edges are we looking for? We are looking for the edges that we can infer through the precision matrix, that is the inverse of the covariance matrix we were talking before. So in this case, this is just an example really. Here, no, it's here, sorry, the pointer, okay. So this is the matrix, I don't know if you can see the pointer. And in this graph, what we have are just the edges that correspond to the direct interactions. So we only see the elements that are non-zeros. Okay? So the interesting property here is that these also express the conditional independence between the variables. What does that mean? So if you see here, for example, x1 and X5, they are linked because they're connected to X2, but they're not directly linked. They are related because they're related to X2. So if we condition on X2, they will be independent, okay? And this is also known as Markov property, okay? And this is indeed a property associated to Markov random fields. There are other examples in this graph that have the same kind of conditional independence, if you see. Okay. Another important concept that is associated to this structure is the partial correlation. So you might read in papers about the precision matrix or about the partial correlation. Indeed, the two concepts are linked through that formula. So the partial correlation is actually derived from a standardization of the precision matrix. So if you look at this example here, so this comes from a paper that was describing psychological variables. I just extracted this very small example from there. So you have here this variable. One is fatigue, concentration, and insomnia. Here we have the partial correlation matrix. And you can see that we have a connection between fatigue and concentration and a connection between fatigue and insomnia. And that's why we can measure a correlation between the two. But actually, that is a latent variable, okay, that we should factor out. And actually, what we should look at is this partial correlation. So clearly, we can see that there's a zero partial correlation, okay? And this is another example where you can clearly see the conditional independence. And you can also see that something that wasn't zero there is zero here in the partial correlation. So this suggests that actually, when we want to measure the conditional independence, we are expecting more sparsity. Okay. Yeah?
How did you go from correlation to partial correlation?
So I am actually trying to link the partial correlation to the concept of, to the inverse of the partial correlation, that is the precision matrix. So
you said that you pose the assumption that there is no dependence between concentration and insomnia given fatigue, right? So how do you set theta 1,3 to 0?
I didn't set it. It just comes from the direct calculation. So you start from, so you measure correlation, then you invert the matrix, and that will give you the precision matrix that expresses the conditional independence or conditional dependence. So you start, what you measure is the correlation, and then you move and say, well, is there any latent variable that I would like to take out of my picture and actually measure the conditional dependencies and independencies actually between
the Gaussian distribution to factor out.
we use it.
On this particular
So no, I'm not using the Gaussian. So you will have to set a model for your data, okay? And then, so let's go back then. We set a model for our data, and we said, okay, the general assumption is this. Well, actually, we will always assume mean equal to 0, but that's another story. But that's the model. In that model, we might think that we want to look at the correlation, right? But actually, we want to focus on the inverse of that correlation, because it's got this magical property of expressing the conditional independencies between variables. So actually, I want to avoid to have a, sorry, a sec. I want to avoid to have a full dense matrix that is that one, because that will capture correlations that are not actually the dependencies that I'm after in my data, because I want to see things that are conditionally independent. That means if I condition on, so let's stick to the example. So if I was measuring correlation, I would think that there's a link between concentration and insomnia. But actually, they are linked through fatigue. So if I marginalize out, I can see that actually concentration and insomnia are independent conditionally on fatigue. Is that clear?
Condition on the confounder at the 0 curve.
Yeah, that in the context that we are looking into is the neighbors of our variables. Okay? Pleasure? Okay. I'll try to make it clearer. I've got other examples, okay? So the idea is that we want to focus on the inverse of the covariance. So let's say just the precision matrix. I hope it's clear what we're talking about now. But actually, we are interested in the support of this matrix. So what we want is just where this is non-zero, because that will express the links, and we will work with a graph that is undirected and unweighted. So is there a link between the variables or not? And we know that that direct link will express conditional independence. Okay, let's give an example that's more mathematical, a random walk. Okay, so assume that I have an IID sample from a normal 0, 1, and I observe these 5 variables, xi's, that are defined like that. So it's a random walk. Each one depends on the previous. So with a little bit of algebra, really, you can compute the covariance matrix there. Okay? So you agree that's the covariance matrix. If you do a little bit of calculation, that's what comes out. And what is the graph that corresponds to this covariance matrix? It's fully connected, right? Everything is connected to everything. I'm just expressing the support of this sigma. But actually, I would like to look at the conditional independencies between these variables. Can you guess what is the precision matrix? So in practice, I have to invert that matrix. But can you intuitively understand the structure? Any guesses? Yeah? Perfect. It would be a 3-diagonal matrix that is very sparse because that is a random walk. So that would be this property of each variable depending only on the previous one. There's like no other memory. And the graph, actually, of conditional independencies is very different from the previous one. This is a fully connected one, a very dense structure. Simple, but very dense. But actually, if I only want to focus on the conditional dependencies, I have a linear graph. Right? It's much, much smaller, simpler structure, and that's what actually I'm after. Okay? The case can be more complicated. I might not know in which order, for example, that walk was taken. I might infer also that. So, about sparsity. We want to study a Gaussian graphical model where the precision matrix is sparse. And this means that when there's sparsity, there will be more conditional independency between our variables. This is our assumption. We want to study this kind of problems. And having to estimate fewer elements of a matrix means that we potentially need less data. Okay. Now, just a bit of an observation. When we talk about sparsity, that would imply that the maximum degree of our graph is smaller than the actual dimension of the problem. Okay? Now, is this a good assumption? Well, it is reasonable in many contexts. For example, in gene interaction networks, we might expect that not everything depends on everything, right? So we expect that the conditional dependency graph between genes is sparse. And this is just from a paper, just to give you an example. So there were 15 cancer samples and 10 normal samples. What they did, so this is RNA-seq data. What they did, they just focused on the genes that were highlighted in cancer pathways as related to cancer. And then only on this list of genes, they extracted the networks, the gene interaction networks, and then they compare all these networks looking for a consensus, and this is the network of strong cross-cancer interactions. So they look for repetitions in the connection of the genes, and that's the consensus. So that's the way that they decided to proceed, and then we will see if we have alternative approaches that are as well valid. Okay, just to give you an idea of the kind of applications. Now, however, sparsity might not be a good assumption, but then we can appeal to the bet on sparsity principle of Tibchirani, and there's no procedure that will do well for dense problems. You might as well just estimate a sparse one. You know, it's your best bet. So it is better to work with estimations in the case of sparsity. And at first, I mean, there's been many approaches to solve this, the problem of the inference of this precision matrix. And one of the most famous is the graphical lasso. The package is called GLasso in many platforms. And what they do, they just solve the maximum likelihood associated to the model, and they add a penalty here, L1 penalty, and this is done to ensure sparsity. So the sparsity is ensured with this. Now, there's nothing wrong with this approach. Also, it has a computational cost of n to the power of 3 and computational space of n to the power of 2. The only problem with this is the independence assumption. Means that features and samples, let me call them like this, just because we have a data set. Imagine we got experiments like single cell data, and then you got genes. So we call them samples and features. They are assumed to be independent. So the observations of the features are taken, consider that the samples were independent, and the observation on the samples are taken, consider that the features were independent. So we can only proceed in one direction. So if we have the independence assumption, then we estimate the gene networks, for example. But then if we want to flip the problem and estimate the network along the other axis, for example, the samples, like single cells, for example, we are contradicting ourselves because we were first assuming they were independent, and now we estimated the dependencies. So we can't really do that. Or we can, people do, but that's wrong in principle, because you can't say they're independent and estimate the dependencies, right? So you can only proceed in one direction. So there's been various approaches to remove the independence assumption. Okay. So we want to have this graph estimation without the independence assumption. But again, looking at features and data points, we could assume the independence of features or data points, and this is a model choice, okay? But we would like to avoid that, so we are looking into a general framework that models the conditional independence relationship between features and data points together. So we want actually to estimate a sparse graph that interrelates features and data points. So we want to estimate a big, let's say, precision matrix that joins together the precision matrices along the two axes, samples and data points, okay, and features, sorry. So, for example, genes and cells. And how is this done? Well, just to understand this, just let's look at two examples. So why we need to do that? So this is a rotating image. It's taken from this paper of Kalaitis at Altri, 2013. So you see there's the duck rotating. Those are the frames. You can decompose the video in different frames that are one after the other. It's always the same duck rotating. So you should not have different networks, right? If you want to think of the network of pixels. But you can also estimate the dependency between the time frames, so that's another network. Or, more, I guess it's more intuitive in this audience, the cells and genes. You see, we want to extract the conditional independence between genes and cells, and we want to infer the two networks at the same time. We don't want to decouple the problems, okay? So you can think of wanting to build a big precision matrix associated to this problem that will put together the precision matrix along the cells and the precision matrix along the genes. So how do we do that? Well, in this paper, there's the link in the slides if you want. It's called the Big Graphical Lasso. They propose to put together these two precision matrices that are here in the, you can imagine those are these two graphs, with an operation that's called Kronecker sum. Now this Kronecker sum translates into the Cartesian product of graphs adjacency matrix. Maybe should have been matrices there, but anyway. So this makes sense. It's very intuitive because if you think of this is a sparse structure, right? It keeps the sparsity and preserves the sparsity along the two dimensions. And this is the mathematical definition. So it goes through the Kronecker product here. And that's the Kronecker sum expressed in terms of two Kronecker products. Now, without going into much of details, you can imagine that those are, we could have used, let's say, a Kronecker sum to interrelate them, but then we would have had a very dense precision matrix that for sure it would not be sparse. And this is able to capture enough variability in the data and preserve the sparsity. Now, what are the limitations of this approach? So even if, in principle, what we are trying to estimate here, if you can imagine to apply to separate graphical lasso in this direction and in this direction in the same algorithm, even if you are trying to infer n square elements here and p square elements here, the way the algorithm was implemented actually implied the memorization of this big matrix at every step. And also because there's a penalization factor here, there's a L1 here and L1 here, because there's the sparsity, we have to impose sparsity, so there will be this penalization, this penalty. That algorithm is iterative, okay? And in every iteration, then an estimation of that big matrix is stored. So this not only has a large a large space complexity of n squared times p squared, but it's actually very slow because you have to do it all the times in your iterations, and then depending on how long it takes to converge, this can be very challenging and unpractical. So you can't really run it on an example that you might want to examine. So what we did, we worked on that with CIGA, and we made it a little bit better, meaning that we worked on the a special way of decomposing this Cartesian product through an eigenvalue, eigen decomposition, and we managed to just memorize n squared plus p squared elements. So we went from this space complexity to this space complexity. It was a big step that implied we could actually work on an example of 182 cells and 167 genes, but again, this is still an iterative procedure because we still have, we're imposing the sparsity, okay, with the L1 penalty in both the directions, both the axes, and so that means that that time, that complexity, that matrix has to be estimated for every iteration. Now, there was an improvement, as what we were saying. We scaled a lot on the memory. And also, there was that this implied an improvement on the computational time. We went from tens of samples to a few hundreds, but this was still not good. So, we looked at other approaches that in the meantime that were developed. Teralasso, Egg-Lasso. They did have a better space and time complexity than Big Lasso, but they're comparable to what we did. So alternative, but not better. And could run on a low thousands. Well, actually, not. I mean, they could run. In some cases, they managed to run, depending on convergence, let's say. But still a lot to do. So the limitations of prior work are that it's not scalable to millions of features as we need. They all present iterative algorithms. In every iteration, there's the need of an eigen decomposition. That means that the runtime for all those approaches that I was showing you is n to the power of 3. And the memory usage is n to the power of 2. In here, I'm assuming n and p are the same, okay? So, but do we need to worry about this? Do we need to scale to millions of samples? Is it really needed? So that's always a question to ask yourself. Why do I do things? Well, if you go to for example, to the single cell expression atlas, you can see experiments that have got about 500,000 of cells. So there's a lot of cells in the same experiments, right? And if you go to the human cell atlas, you got here 4 million, 2 million, almost 2 millions, almost 2 millions of cells. So if we want to analyze these experiments in one go without pre-selecting the cells we want to work on, we really need to work on the methodology development so that we can handle that, right? Also, this is from a paper of 2021 from Krachenko, and it shows, let's say, here on the red line, sorry, I can't see the point, there we go. Okay, on the red line, you can see all the different platforms that have been developed during the years, till 2020, more or less, and there's a log count of the cells that could be sequenced on these platforms altogether. And that is the same log count of the number of transistors on processors, okay, on computers that we need to use to analyze the data. And so that trend has respected so far the Moore's law. That means every 2 years that number of transistors doubles. And you can see that in order to handle that enormous amount of cells, we really need to improve the methodologies. There's the need to do something because we can't really otherwise analyze this amount of data despite the increase in computational power, let's say, that we have. Now, so the answer is, do we need to scale to millions of samples? Do we need methodologies that enable us to do so? The answer should be yes. I hope I convinced you. So that's where I would like to talk about the other approach that we have developed recently with Bailey. This is here. And we'll guide you through some technical parts and examples later. So this approach is a fast multi-axis Gaussian graphical model. I will talk about this now. Sorry, it's not working anymore. I don't know. Okay. So let's consider first a little bit more on the kind of examples we want to work on. So we have discussed up to now like data sets where we could see one tensor, like cells and genes, for example. We can also have multiple tensors, or in this case, multiple omics. That are the different modalities we refer to in these examples. So, for example, you can think of metagenomics and metabolomics. You have 2 axes per data sets. One is people and species. One is people and metabolites. You can see that in this experiment in particular, those are the same people. And that's why when we plotted this, we kind of exemplified the same graph, because these should be the same. So we should not decouple samples, okay? As we didn't want to decouple, let's say, and consider these dimensions independently, we should not consider axes that are shared independently, because those are the same people, okay? And our approach not only addressed scalability, but also this problem. So, just from a modeling point of view, the tensors, so that means the different modalities, that are sharing axes will still be drawn independently from these Kronecker sum normal distributions. Okay, so you can imagine precision matrices along each dimension, and then that would be the big precision matrix that is given by the Kronecker sum of all of them, okay? But we have to pay attention of keeping the same precision matrix on the shared axis. That will create the dependencies between the tensors, okay? That will ensure that we are actually estimating the same graph on that shared axis, okay? Now, what is the real improvement in our approach to GMGM? So, as I was saying in previous work, the big issue was that the sparsity was imposed with this L1 penalty. And that implied iterative algorithms. And that implies that at every iteration, you have to redo an eigen decomposition. And that means that for every iteration, you have a runtime of n to the power of 3. Now, the main idea is that if we remove that regularization, we will only need one eigen decomposition. And then if we ensure the sparsity some other way, we can then reconstruct our precision matrix. So in place of regularization, we used thresholding. And that was, that is Bailey here. Okay. So is our approach much faster? So this is synthetic data, runtime, and you can see that on a data set of about 5,000, we can run in about 60 seconds. While you can see previous approaches, runtime, and we cannot actually even run on this amount of data. And on real data set, GM can also run of 10,000 cells, for example, in under 1 hour. This is how it scales. Okay. Now, is our approach good enough? So we do still hit the memory limit of n squared, okay, because that is actually optimal for this problem. We can't avoid that. And how can we tackle this limitation? We will need to have additional assumptions. Okay. Now, these additional assumptions are actually part of an ongoing work that is unpublished, but the software that is available on GitHub already includes these updates and is available. So the additional assumptions we're working on are considering a low-ranked version of both the input data set and the precision matrices that we want to estimate. So the idea is that we are now, at the moment, in the eigen decomposition, considering all the eigenvectors, okay? But we might need less. So this is equivalent of considering a certain number of principal components of your data or of the estimated network. That precision matrix, exactly. That's what we are doing. We are working on a low-ranked version based on the principal components of the matrices, of the input one and the one that we want to estimate. Also, to tackle the memory, so that kind of works well for the computational, for the runtime, while to tackle the problem of the memory, now we also ensure the fixed upper bound on the output graph sparsity. That means that we have in mind how many non-zero elements we want. That will not be, if that is fixed, it will not depend on the dimension. That will not grow linearly with n. Okay? And in our faster version, let's say, and the ongoing work version of GM, that's the results. We have a computational a computational complexity of n squared, and then we have a space complexity of n. And this is the projection of the times in seconds. So you can see that with the 50 principal components, and we kept the same in the algorithm for the input and output, we are this low in terms of runtime. That is a huge improvement. Of course, this comes with some observations. If you have a small data set, you should run the version that doesn't do all these approximations because it would be much more precise. And we do not have results to show here today about this, but we observe this as expected. If you have less data, you're better off having the original GMGM that doesn't do all these assumptions, and you will have much more accurate results. But when you have a lot of data, you get away, let's say, with all these assumptions, and you have a reasonable estimation, and your decision is between do I run something or do I run nothing, because you can't really run anything on large data sets. Okay. So about results. So we show some results on a data set, just to exemplify, let's say, the run. There is immuno-landscape of viral and carcinogen-derived head and neck cancer. So this was the paper, so we took the experiment there, and from single-cell expression atlas, and there were 157 cells and 25,000, about 25,000 genes. Now, how did we compare to prior work on this? So we only needed to use the fast version, okay, of our algorithm. Only needed to use 2 gigabytes, or gigabytes, sorry, gigabytes. While prior work would have needed, this is a projection, of course, 440 gigabytes. That means you really can't run prior work, okay? But you can run our approach, and you need this amount of memory. About run times, we run in 20 minutes, while prior work would have needed this projection is based on the runtime that we know, 2,344 days, okay? That means, again, you can run it, not only for memory use, but also for runtime. But do we do well a million cells data set? That was the initial question. Are we yet there? Well, unfortunately, we still have some limitations that are physical limitations that we reach on Bailey's computer, because even when the data are highly sparse, a normal computer, a good one, has got up to 8 gigabytes of RAM, so you will need something larger. For example, on million of cells, you will need, with prior work, 16 terabytes, but depending on the sparsity that we impose, we will need about 20 gigabytes, and potentially we could do it, but we need a more powerful computer to do so. And the prior work would have run in 1,901 years, and this would run in about 15 hours on a computer that has at least 20 gigabytes of RAM. So, back to the example, some applications. Okay, so the data set I was mentioning, where there were genes and cells, so we run on the overall data set without pre-selecting cells or genes as a bunch, and we then extracted the networks along the genes and along the networks. So ways of validations, what we did, we focused on some of the modules of the networks, for example, on genes, and then we investigated the GO-term associated that were more significant, and then we went into the string database, and so which were the proteins that were coded in association to these genes, and we saw that those were the connections. So these are just examples. We're not claiming that we have any results on this data set. It was just, we were just trying out on this data set, and those are possible ways of proceeding for validation of modules And we open to discussion on how to do this when you are exploring a data set. This is another module. We did the same kind of thing. So this is the module. We are just showing top 25 genes by degree. And then this is the corresponding string protein network. And those are the Go terms associated. And then we also want to show something that was a little bit strange. So this module, for example, we didn't discover much in terms of Go terms. But then when we looked at the protein network, they don't look associated, but the names are all similar. That makes us think that they are related somehow, but even if there's no known association. So the exploration is all about, or did we find meaningful relationship? And that's the answer we're trying to find when we look around for, let's say, validation of the results. So all up for interpretation there. And then, oh, sorry, this is repeated, sorry. Then cell clusters. We looked at the cell network, and we found, with the Leiden algorithm, 23 clusters. Now, on the left, we have a plot based on the UMAP projection and the t-SNE projection, and we are overlaying with colors the 23 clusters, okay, that we found. And then we wanted to validate these clusters, trying to identify the group of cells if there were any relevant cells. So what Bailey did, he went through some literature and tried to find marker genes for specific groups of cells. So he identified some marker genes, then went into the clusters and looked at the median expression of those genes. And the darker, the more expressed. Okay, and so we might guess that we have a kind of good result here or here. So we could imagine that maybe cluster 16 has got a lot of myeloid cells, or same thing here. So that's the way we decided to proceed. But you can imagine that this is heavily related to what other people have found out in other papers, and also related to the ability of us finding and identifying in these papers markers. The conclusion that we have is that we can do all of these, but then when there's no ground truth, you really need an expert to help you understanding the results, because I mean, you need someone to tell you this looks right or wrong based on my knowledge, and again, any suggestion is welcome. And I think that other than that, we could be ready for the code space part, but first, I'm happy to take any other question. Yeah. Yeah?
Thank you for the talk. I have some questions on the practical perspective. So it seems that you talk a lot about memory. So I think this indicates to me that you're running it on CPUs, right? Have you tried like using a GPU?
We didn't use graphical processing units, no.
We've not done it on a GPU, but I don't see how that would solve a memory problem.
No, it won't. It won't. It's just going to be much, much faster.
Like always gets a computer with a terabyte of RAM.
And the second question is kind of related. So I think one of the major problems that you have is like estimating the eigenvectors, right?
Mm-hmm.
It's a hard problem. But I know that there are certain classes of matrices, like for example, the ones that they use in engineering for finite differences, where it's a very particular example, right? But you get your eigenvalue and your eigenvector for free, right? As soon as you define the matrix, you can work them out before you start your computations. would there be a way to transform some, like, by imposing certain assumptions to the problems, to transform these matrices into some well-known classes where you can get easily some of your eigenvectors and do that?
I mean, I think.
Essentially, I'm trying to suggest that you probably need to impose a lot of.
You want to answer? Yeah. Okay, Billy wants to answer.
So it'd be great if we could do that. I think that would be really useful. I think it probably won't be possible though, because the eigenvectors turn out to, they can be the eigenvectors of any positive standard definite matrix. There's no, in the general case, there's no restriction that I think we could use. Sorry if I misinterpreted your question.
No. I mean, you probably, I was thinking of what if you can impose some assumptions which will lead you, not necessarily this sort of, this set of matrices where you can get things for free, but if you are close, then something close.
I mean, I think you can do it, but now then there's a lot of exploration to do, understanding what you are restricting in terms of then of the discoveries, because that matrices will give you the conditional dependencies, and of course, you imposing too much might make you miss out, or maybe just focus on specific problems. But it's a very good suggestion. We can look into other kind of constraints. Is always a good thing.
I mean, you might have something iterative, right? So you could start something which is like imposes a lot of assumptions, and you get to a very sparse matrix, and then you can slowly, like, start separating your graph, and then you can slowly doing it more locally, and then I don't know, something like that. But I think there must be some way to get your eigenvectors for a relatively free. Necessarily, but there are certain, I know certain matrices can help you do that.
So you're right, but I think at the moment the physical limitation is just actually also downloading the data.
Well,
so the data we need to be downloaded. They're just enormous, right?
Yeah, we've again hit a memory limit in terms of just like I think the main roadblock to further scalability is now the memory again. But you are right. I think that this would be.
Yeah.
I think that it would be a really good thing to do.
Well, I mean, if you're running it on your laptop, yeah. If you're trying to do big data in the lab.
Well, that's the dream. Anyway, okay. Other question? Yeah.
So you did a lot of approximation to reduce the runtime, but also have you quantitatively measured what is the cutting performance?
Can you repeat? Have you measured?
A lot of approximations and assumptions in order to reach off on n, and you never showed like a quantitative.
The accuracy, you mean?
Sorry?
The accuracy?
So you did the approximation of your runtime so that you can scale it to off on n, and you only measured the time, but also what is the accuracy?
That's a very good point. We were discussing if showing the accuracy result or not, but we do have a kind of trade-off. But you can just say more. But in practice, we suggest to use this kind of approach when you have a very large data set. On small data sets, you're better off not approximating this much, because you can still run. You do not have big limitations in terms of computational time and space. And you're still fast enough with the original GMGM, I mean, and it is as accurate as previous approaches, but just can run on larger data set, but not that large. Yeah.
What I think would be, for example, fair is to, let's say, sample few of the points of few of the cells from the 2,000 million data set, run the expensive algorithm, then run your algorithm on whole cells, and then compare the accuracy.
So the thing is that we can only, so in order to test the accuracy, the best approach is to work on simulated data, and we've done so. Can you?
I can tell you what these, because on simulated data, what we found is that, let's say you're looking at area under the precision recall curve, just because that's the only thing I have in my head right now.
Yeah.
If you're on, let's say, a 1,000-cell data set, and you're working with one of the firewalls, then you'll get quite good precision recall, and you get about half as good if you use our algorithm with very stringent restrictions, have big assumptions. But when you scale that up further, the accuracy gap closes quite quickly. So once you're looking at 2,000, 3,000, 4,000 cells like that, it's even if you keep the same assumptions, it's closing in on that. So when you're at 100,000 cells, obviously we haven't been able to compare it to prior work, but assuming that trend holds, you should be having area under precision recall close to 1. Based on
you can use area under the precision recall curve only if you have positive and negative classes, and in single cell you have multiple cell types.
So on this simulated data, we're doing did it get an edge correct or not? So if that's a binary.
Maybe you can look into this well-known and annotated single cell data set like PBMC. It's just 10,000 cells, 20,000 genes, and look there. And also one approach would be have you tried node to vec, where you have your graph and you would like to end up with vector representation of each node.
We have not used node to that.
Yes, because the runtime of node to vec is also n log n, and it also scales on GPUs.
Yeah, but it won't give us a precision matrix. It won't give us a graph with the interpretation of a conditional dependency graph.
Yeah, but maybe it's a proxy. So
Yeah.
if you already have the graph structure in the vectors, then it's a good proxy.
Sure.
Can I ask
Yeah.
about the simulation? So is it getting better because there's more data to estimate the covariance of the genes in that example, as you add more cells?
So yeah, I think that's, but even if we are expanding both dimensions at the same time, so in simulated data, if you had going from 1,000 genes to 10,000 and 1,000 cells to 10,000, then it's still increasing. So
Yeah, but you've still got more data per link. But,
yeah.
but I suppose in very heterogeneous data set with lots of cell types, the number of cells per cell type could still be small. The atlas may be growing, but the number of clusters of cells might not be so big. So the estimation of the correlations within a cell type could still be not as, so it might not be as good as that because
That's true.
the heterogeneity and the size might grow at the same time with an atlas, I suppose.
Yeah, that's a good point.
That's true. I think we should work more on the simulation settings, simulating different scenarios. The thing is that now the very last part is the ongoing.
I'm running it.
Okay.
I'm running this.
Great.
Yeah,
Okay. So, but definitely we are looking also into aspects of data simulation because, of course, when there's different ways of doing it, and it's also a challenging problem. Simulated data where you know the dependency structure when the dependency structure is more complex, like having more tensors. Also, simulating data is a problem because you should fix the dependency structure and then generate the data, and it's also very expensive. Let's say it's hard. So we kind of focus on simpler examples, but you're absolutely right. Yeah.
For modestly large simulated data sets, it takes longer to simulate the data set than to run the algorithm, which is quite annoying to test how it does on larger settings.
Yeah. Yeah. Yeah.
Are there any asymptotic results for as n or p goes to infinity on this? Because it seems like the graph you're trying to learn is kind of growing at the same rate as the amount of data you have to learn the graph with. And I'm not clear if there's are you able to actually, if you had infinite data, exactly recover the positioning matrix?
So there are asymptotic results. I don't remember the exact rate off the top of my head, but there's the paper for Teralasso, one of the prior work algorithms, by Greenwald et al. They have, like, they give good asymptotic results for how it recovers, and it's very similar to the asymptotic results for just the normal graphical SL. There's like a log factor added in there, but otherwise it grows, like it approaches correctness at a similar rate.
Yeah.
Yeah.
But it's a very good point. It's something that we should specifically address. How many data do you need for a good estimation? That's also another point we didn't discuss. Yeah. Yep.
How do you select the regularization threshold?
We don't. So we decided not to go for regularization. In the end, we decided to select the number of eigenvectors. I mean, do you mean in the latest version that we were discussing about? Yeah. So we just decided the number of principal components we want to work with. But of course, that is still we're testing it. We have, on the example that Bailey has been running, we set around, they were pretty big, we set around 50, but again, it's as arbitrary as setting a threshold. Yeah.
And well, so you have to set a threshold for how many edges to keep
Also,
in
yeah.
the end as well, which is what directly controls sparsity. And just like with regularization, it can be hard to figure out, especially on real-world problems where you don't have easy access to validation sometimes. It can be hard to figure out how to set those values. So we normally go with like a quite conservative estimate of like keeping more than we think we should, because we can always then threshold later, but I don't have a rule of thumb for you, unfortunately.
I mean, also with the regularization, it was a bit of the same kind of things. You work on simulation, you try intervals for your parameters, and then you kind of have a guess of what that should be. But it's a big question mark also there, like, you know, every time you have to take a choice. Yeah.
Just picking up on that asymptotics, a long time ago I worked on random matrix theory, and in that case, if the dimensions and the number of samples grow proportional to each other, but the noise per row stays the same, the situation improves as the whole matrix gets bigger.
I see.
Because the asymptotics are a bit weird for random matrix theory, and they basically force you to increase the noise as the size of the matrix increases. It's a really weird result. So sometimes you can surprisingly learn the eigenstructure of a big matrix really well in that limit on real data, where every gene is like a new observation with IID noise. It's the same as the other gene. So it's like an interesting asymptotic area. So like the situation might get better as everything gets bigger, but that's not in the case where you have lots of different components. Thing, which might make things more complicated. So you probably have to simulate that.
Yes, I mean, there's a lot to explore. You know, at the moment, we're just happy that we have something that actually can run and can handle a large data set. Now, how good that is and how good it can get and how can we make it better, I mean, still, there's still a lot to work on. But at least, you know, we progressed. You know, that's okay. So, any other questions? So, yeah.
We can go on to the GitHub code base.
Yep.
I was wondering, on your point about using the principal component analysis, is that do you literally do the principal component analysis on the data, or is it just you're only finding the first few eigenvectors? And I only ask because if you are doing the principal component analysis on the data, have you considered using fancier dimensionality reduction techniques that could possibly capture more of the actual underlying structure of the data while maintaining fewer data points? And does that kind of break the maths in some way?
So it does break the maths, but I don't know if it breaks the maths in a way that actually becomes a problem. Like, I think it would actually be kind of interesting to try it and see if it works anyways. But the reason why we say it's like principal component analysis is in the maths, it turns out the eigenvectors of our sample covariance matrix, when you don't have regularization, they correspond with the eigenvectors of the solution. So you eigendecompose and you get it for free, and that's why that's there.
So we need the eigen decomposition.
Yeah. but that's just like finding like the linear hidden space in your original data or feature space, depending on which one you're doing PCA on.
Yeah,
Like possibly,
I think
dimensionality reduction.
it would be interesting.
Yeah.
Really, like that's quite exciting.
So the benefits would be interesting how you structures. So I just wanted to look into it.
Yeah, we haven't, but like I really like that idea. It's quite cool. I think as good.
It's good. Okay, I think maybe we should start on the practical part, otherwise we won't have time, I guess. Okay, so I'll leave it to you.
Sure. Hi, I'm Bailey. You've heard me speak. So if you have a computer and you want to participate in practical, which is hopefully all of you, we have a link up here that I'm going to give you like 20 seconds to get on your computer if you haven't already. And then I'll walk you through how to start the practical. It does involve downloading some data, which might take 2 to 5 minutes. Don't worry, it's not downloading on your computer. It will be on the cloud somewhere. So I'm hoping that you've now all typed in the link. If not, shout. Good. Good. No shouting. Oh, that's not the mouse. This is the mouse. There are 3 mice up here, so if I get confused, do forgive me. Bring this back up. So if you click the link, you should arrive at a GitHub repository, and it has some instructions on here, but I'll walk you through it. You should have a GitHub account. We already asked. Oh, yes, thank you. This would be great. I don't really know how to put it on. But yeah, I might have to take off my little magnet thing. Oh, it goes down there?
Yeah, I think.
Oh, hey, okay, I got it.
So it's touching your bed.
Awesome, and then I'll just strap this here. Is that better, or is that too far away from me? It's good? Okay, great. So yeah, you should follow that link to a GitHub. It does have instructions. So what we want you to do is fork the repository, which I'm not logged in right now. I don't remember where the button is to fork that. I'm going to log in and see if it appears. And it might not appear because it's my repository, so I don't know if I can fork my own. But let's see.
If it doesn't work on Firefox, try Chrome.
Okay, thank you. We will see. Oh, I'm quite painstaking at typing into this because you may be able to tell from my accent. I'm used to American keyboards, unfortunately. Yeah. Sorry. Well, the link. Let me go back to it. Let's see. Yeah. Okay. So my name's Bailey Andrew, and the link is because you're at the ELLIS Summer School. So GitHub/my name Bailey Andrew /ELLIS Summer School will get you there if you forget. Yeah. And is that good? Yeah, that's a good point. If you take a picture of it right now, if you haven't already, you have 10 seconds. I'll spare you the countdown though. Okay. Yeah, that seems like it was now going to be about 10 seconds. I keep touching that mouse. Let's get that over there. And let me log in.
The link is now also in our Slack.
Oh, 2-factor authentication. Yep. Yep. Oof. There we go. Okay. Sorry about that. I am now logged in, and I'm not going to save my password. So yeah, there should be an option to fork the repository. It might be in this little green button. Maybe it's not. Oh, yes. Yeah, there. And it's grayed out because I'm not allowed to fork it, but you are, so please do. I can tell that presumably 8 of you have, 9 of you have, because I know Luisa will have forked it in the past. So once you've done that, there's this green button, code. There's a little place called code spaces, and you probably don't have code space on it. So you'll click that button, and it should take you to something that looks like this, which can be a bit slow to load. So
we'll see what happens. Okay, it's loading. So what I want you all to do is go into notebooks and raise your hand if you start like falling behind, if I'm going too fast on the directions. But I think it's probably me that's the bottleneck here. Go to Setup, and once it loads, just click Run All. And that will get all the packages and data downloading in the background so that I can then tell you a bit about the data set. Hopefully, once I'm done telling you about the data set, which should take maybe 2 minutes, then it will have downloaded on your computer. So if you haven't clicked Run All on that, then let me know. Still setting up. Okay. In the meantime, I'm going to try to find the paper link and go ahead and prepare to start talking about it. So again, if you have got the code space, then you should go and open up the setup file inside the notebooks folder and just click Run All. And so either you should be hopefully waiting for that to load or waiting for it to run all. So in the meantime, I'm just going to talk to you briefly about what this data set is. So it's from a paper called Spatiotemporal Immune Zonation of the Human Kidney, so that you can imagine it contains immune cells and maybe cells you find in a kidney too. There's about 30,000 cells in it. This paper says there's 40,000. It's in the Human Cell Atlas. I don't know about you guys. I find whenever it's a Human Cell Atlas data set, you can find many different reports of how many cells are in it, which is kind of annoying. I think it's because they'll probably just keep on updating them, though I don't know why the amount of cells has gone down. But yes. And it has multiple people, at least 20 people, unless they've taken 2 kidneys from the same person. And they've already done some clustering on it and identified cell types. So there's a lot of like clinical information in the data set and their estimation of the cell types, which it has been done using clustering instead of cell by cell. Now, it may be worth, if you've been waiting for the code space to load, for you to check and click Run All. I don't want to right click. And yeah, let me just quickly talk about then what the workshop will be. And then finally, everything should have probably loaded on everyone's computers and you can get going. So I apologize for drawing this out, but I don't know how long everyone's computers are going to take to download it. So this is a workshop. It's going to look at how to analyze this data set with 2 algorithms, just the original graphical lasso and then the GMGM algorithm that we've been talking about here. So the graphical lasso, it's all in Python, so hopefully you're familiar with it. I apologize if not. I have found that the graphical lasso implementation in Python can be a bit finicky compared to those in R. So we've put everything in Python because it would be really confusing to make everyone work in 2 languages. But if you're going to use the graphical lasso, I would recommend using the R versions when you're in your own work. And what we'll look at in this workshop is just like choices for pre-processing. There's lots of opportunities in the workshop where you can play with the values that we chose. So a lot of the values that we chose were just arbitrary, especially for pre-processing. But then we encourage you to, once you've run the notebook, which shouldn't take too long, you can then play around with them and see how that affects results. Almost done. Then we look at different ways of validating the results. So looking at the gene graph and like, sorry, it's hard to scroll here. And like seeing how they might be connected in your output graphs for, like, for example, this is a heat shock protein. So you would hope that maybe they're connected because they all seem to be part of a similar class of genes. And you can compare different methods and what they find. And then also looking at the cells. I think I'm going to stop trying to scroll through it because I either go too fast or too slow on this computer. Yeah, and you can also look at cell graphs. So we have different ways of validating it. I'll let you all get on with the workshop, try to play around with it, see what happens. If you don't feel that confident in like coding yourself or messing around with it, then just clicking Run All, you should be able to, like it should just run all, and then we have some points where you can change values and see what happens. If you're comfortable with coding, put in your own methods of validation that you use in your own work and see what happens. Yes, has everyone, has the data set downloaded for everyone? Okay, so I see some yeses, some nos. If it has downloaded for you, just go ahead and start. If it hasn't, sorry, there's nothing I can do. You just have to wait till it downloads. And if you have any questions at any point, raise your hand and I'll answer them. Until then, I'll probably stand around here awkwardly, maybe pace a bit, or sit down. Enjoy.
I'll ask you a question.
Yeah.
I guess throughout the whole method, I don't really understand the meaning of like a person-person interaction network or a cell-cell interaction network. So I guess that network somehow influences the network you infer for the other stuff, right?
Yeah. So if you mention a person-to-person interaction network, so we can imagine with like bulk data, like bulk RNA data, maybe people in the same family are in the data set, so they'd be connected, or people in the same regions. You might not be interested in the results of that, but like you say, the dependencies there actually do affect the dependencies you find in the output.
So they're correlated somehow.
Yeah. So even if you're not interested in that graph, taking it into account will improve your results on the other graph. Now, if you're working with single-cell data sets, you might be interested in the single-cell graph because you can cluster on it and hopefully you see like, oh, all the B cells are clustered here and that. Yeah.
It should be in the same model anyway, because it's more correct.
Yeah.
You know?
But then I guess you have to be kind of meticulous about choosing where you fix the interaction network of one of the two networks should be the same across different. Did you have some examples of humans and
metabolomics and metabolomics?
Yeah. And so you're right. If you fix them across the two like modalities, that's not always the best thing to do. You might expect things to change. The main, at least the ways I'm aware of to make sure you can make them similar but account for change, involves regularization, which is the bane of scalability in the multi-axis case. So that's why we haven't done that. We've just considered when there's exact overlap. But a good avenue for future work would be to, yeah. Yeah. Has it what? Sorry.
So for your like large scale,
yeah.
You're not doing like an L1 derivation. So you're doing a full dense inference and you're sparsifying it with your threshold.
So kind of like on paper, yes, but in implementation, you can just, let's say you want to keep the top 10 edges overall. You can construct the output like edge by edge and just keep a running list of the top 10. So you never actually have the whole thing in memory.
Oh, I see.
Yeah.
Another question.
Yeah.
Have you, I guess you might already be doing this, but are you doing any sort of like randomized algorithm for getting the, let's say, the principal going faster or something?
So with single-cell RNA sequencing data, because it's highly sparse, we're just doing the sparse singular value decomposition routines in the Dask library. I don't know if Dask. It's a way to do like, instead of loading things into RAM, keeping things on disk, but efficiently. That's why we're using that.
Is it randomized or is it?
I don't know the details of their algorithm. I did.
You know, there are some algorithms.
Yeah.
Let's say you only want the top singular vector
Yeah.
or singular value. You can obtain it really quickly.
Yeah, that would be good. Yeah.
If you just look into like randomized SVD.
Okay. Yeah.
It's a huge thing.
Yeah, that's good. Thank you, because that takes 80% of the runtime of the algorithm. Is the eigenvectors at the start?
Yeah. So
Yeah.
You don't need the full thing. If you just need the top 5, it's really, really fast.
Okay.
To get it.
Thank you.
Because I do a need this in order to.
Yeah. So it's not, is it the data that's not downloading on your computer, or is it the code space not opening?
It's the setting up the code space.
Oh.
Yeah.
It may be worth reloading the page. I'm sorry that it's seeming to be, yeah. So setup will download the data and then, yeah. And that should just take 2 minutes, hopefully.
About this, not about this repository, but about how you said that this was called a code spaces.
Yeah.
I think maybe I want to use it myself in the future. So when I was running, this I haven't been using this feature at all before in my life. So is this reckon we're running with the kernels being run on a remote server or something?
Yeah. Yeah.
Did you buy that?
No. So you get, I think you get 40 hours for free, like 40 hours per core. So like yeah. So I've used up hours faster than I thought. Yeah. So the 40 hours for free per month and
per month per account.
Yes.
But it doesn't mean that if someone attends 2 summer schools consecutively, whereas it may run out. Okay.
So the reason why we chose this over Google Colab is because this, when you download the data, that's actually persistent. So even when you turn it off, like it stores it forever, and you're not using hours.
Yeah. Even if I'm not using it for a prolonged period of time, it'll be stored in GitHub, I presume.
Like on their remote server, they still have it.
In data associated with this, would they be put on public domain then if you use a public repository?
I don't know. So like this data set that we downloaded, I guess it's in that folder, that's not stored on the GitHub.
Oh.
But it is stored on my code space. So that's why you all had to download the data. But now it's in your code space. You never need to download it again.
I see.
Yeah,
that's interesting. Yeah, I'm just very confused about all the kind of things. Like,
yeah.
we can say that click a green button or so. What a green button. I just like literally what first thing I do is I just went to git clone and then I listen to what you're talking about.
Yeah, well that was our backup plan if someone in the audience didn't have GitHub, which I know is unlikely. But then we tell them, okay, here's how you download it. And
well, to be honest, I mean, as far as my personal experience goes, the huge amount of STEM subjects in this country still don't teach undergrad students what the GitHub is. Like, literally, when they told me about GitHub, it's like, just use it as a free hard drive, alternative to Google Drive. No one touches what fancy properties of it.
Yeah.
It's their fault. Thank you.
Yeah.
That like the normalizing introduces conditional dependence,
Yeah.
but that violates the assumptions of your model.
Yeah.
Therefore, it doesn't actually matter so much.
No, so the norm. Should I take it off?
No,
You can just turn it off.
just a second. Shall we wrap up because we are at coffee break? Shall we
Sure.
just?
Yeah,
so
I'll
we
let
can,
Lance
can we continue? Just at coffee, and you can say things like, "How did it go? Do you got any questions, comments?"
So I should ask them questions now, or?
Yeah, because there's coffee now,
Okay.
so we should go.
Hey everyone, it seems to be the coffee break time now. So if you want to keep working, you have questions, ask me, but I believe you're free to go. Yeah.
If anyone discovered a new cell type or a new
Yeah.
interaction, then they own it. Right? This is actually what the group all right paper.
But this Luisa Bailey.
Every person has to make